each lab that you do rotate in is a bit different. And a lot of it is in terms of the environment that you're in. So every PI is slightly different, your colleagues will be different, and they'll have different ideas for what they want you to do there. So sometimes it's more practical for you to tag along with someone who already has an established project in the lab, and sometimes you might be starting something new and different. So in my first rotation, I was in the lab of Edward Ruthazer at the MNI, and we studied neurodevelopment in the tadpole visual system. And I started off with a brand new project that no one was really doing in the lab, in an area that people don't look at, which is in this particular lab, which is myelination and how that might be dependent on activity. So it's a wet lab experience where you come in, your hours aren't necessarily 9 to 5, but you're there where you need to be to do the experiments you need to get done. My second rotation was in Dr. David Stellwagen's lab at the Montreal General Hospital, and that was an electrophysiology lab. And I was just learning how to do basic electrophysiology, patch clamping, something that I had never done before. And the idea was trying to characterize interneurons electrophysiologically. And then the third lab that I rotated at, it was at Florian Stork in the Douglas Mental Hospital. And that was a bit outside of these two fields, looking at circadian rhythms, trying to get a sense of how you could study circadian rhythms in a behavioral way.
I came from a behavioral genetics lab at University of Toronto, and I was interested in how the environment interacts with the genome and creates individual differences. And I thought these two labs, the Mini Lab and the Treki Lab, were really good places to explore that in relation to the gut microbiome as one of the environments that could shape our genome. I think it's true with the rotation program that each lab is going to be different. You're not going to know if you're going to have your own project or if you're going to be working on another big project. But I think most PIs are pretty open to what a rotation student wants to do. So in the Mini Lab, I developed my own project. It took their model, that existing rat model of maternal care, and I looked at the microbiome. In the Treki Lab, I worked on a larger project on myelination, actually in the anterior cingulate cortex in the brain. That's being submitted as a paper. It has been submitted. And in the Lueshi Lab, I basically just started to work on my PhD question. I started to look at, use mice, look at the microbiome and depression. So for me, I organized a rotation program for myself that gave me different tools that I needed to eventually do the kind of PhD project that I wanted to do.
I rotated in Dr. Leslie Fellows, Dr. Elaine Dagger, and Dr. Marco Layton's lab in that order. So when I first started off, I kind of knew before even coming, because I did a master's, that I wanted to study decision-making, dopamine sort of related things. And all three labs kind of cover that topic in slightly different ways. So with Dr. Leslie Fellows, I was mostly doing a big meta-analysis on decision-making tasks. Then with Dr. Elaine Dagger, it was more for doing it from a neuroimaging MRI perspective. I took some open data sets and I analyzed that data, looking at cortical thickness and correlating with measures of cognition or disease severity. And then for my third rotation with Dr. Marco Layton, I was analyzing some PET data that was already collected as well, and we're trying to write that up as another paper too. So in the end, I ended up choosing to be co-supervised by Dr. Leslie Fellows and Dr. Elaine Dagger, and kind of merging the two together. So taking both the decision elements and then putting in the neuroimaging side from Elaine's lab, and kind of trying to combine that into my own little personal PhD project.
My first two rotations were in slightly larger labs. So the projects that I was involved in was mostly helping senior members, such as a PhD student or a postdoctoral fellows. I definitely learned how to do experiments on my own, so by assisting others, and also got some taste of analyzing the data myself. However, I got the opportunity to manage my own projects. in a smaller lab where I was able to manage the projects myself. So in this case, I participated in, let's say, an optogenetic project where I went all the way from constructing the vectors, the various vectors themselves, to stereotax surgeries into animals, into all the way to recording from neurons, stimulating them optogenetically, and also behavioral testing, right? So the experience I got from sampling 3 different labs, so starting my project again 3 different times. So that certainly helped to alleviate some of the later challenges that I could be facing in my PhD.
So my first rotation is probably the one I have the most to say about because, well, A, it's the longest, and B, it's the one I ended up returning to. Because it was an imaging lab, it's hard to have a specific project because those studies take a lot of time, but luckily there was already data in the lab, so they let me sort of help with the analysis of that, and currently there is a manuscript under review from my time there. In my second rotation, it was an electrophysiology lab with rats, so then I had a more defined project, but I didn't quite have time to finish. I got animal training, I learned how to work with rats and to analyze the data. And then my third rotation was a bit complicated because it ended up being a rotation that was split between 2 labs. And this was because I wanted to learn techniques from those 2 labs as I thought they would be useful later on. So one lab was to learn how to do DTI data, and the other one was to learn how to do EEG. And the DTI stuff is coming in useful now for my PhD project.